So today we are beginning with a new pipeline, that is metabolomics. This is a GUI version of metabolomics data analysis using these two tools, that is MS Dial and Metabo Analyst. So basically, metabolomics is the analysis of metabolites that is present in our cells, tissues, and um, in our body. So collectively forming the metabolome. So study about the metabolome, just like genomics and transcriptomics, we have given a name, that is metabolomics. So just like genomics and transcriptomics, both the genetics and the environment can affect the metabolic substrate and can change the phenotypes and thus resulting in the expressed characteristics. So unlike the other omics, metabolomics directly reflects the real-time biochemical activities. For example, if we are doing genomics and transcriptomics, we are studying mutations, we are studying transcriptions and all. But still, there are so many other processes like splicing, post-translational modifications and all. Even after protein is formed, there it undergoes some shaping by chaperones and all. So basically, the end stage uh, by which the phenotype is determined is the metabolites. So, the study of metabolites is it's considered the most accurate representation of an organism's molecular phenotype. So, there is um, uh, more importance given to metabolomics. So, this is the hierarchy how it comes: genome, transcriptome, then proteome, and then metabolome. Metabolome is the final, the last compound produced in our body that is that defines the phenotypes. So basically, if we take the example of a bacteria, there are two types of metabolites. One is primary metabolites and then secondary metabolites. Primary metabolites, there is essential metabolites like amino acids, nucleotides, vitamins, enzymes, and all that. And then there is metabolic end products, that is ethanol and organic acids. So one of the characteristics that is only found in bacteria is the production of secondary metabolites. Bacteria and plants is the production of secondary metabolites. So there we can see antibiotics, pigments, and toxins that we can commercialize and use it as products. So basically, by doing metabolomics, we are checking for the one type is to quantify like how much of the metabolites are present in a bacteria, or to check for new compounds, or if we get a new species of bacteria, to check whether um, similar compounds are present in a bacteria or not. So just like that, metabolomics have a wide area of research possibilities. So these are the two types of metabolomics. One is targeted and one is untargeted. So targeted means we are searching for a particular metabolite in a species, or bacteria or any organism. So will be, for example, if you are looking for nicotinic acid, we are looking for the specific metabolite pathway related with nicotinic acid and its quantification, how much of the compound is produced in that particular bacteria and all. So in targeted metabolomics, we'll be studying about a particular selected compound. But when it comes to untargeted metabolomics, uh, we will be uh, looking for biomarkers or new compounds. So if it is a newly identified bacterial species, we'll be looking for new compounds which could be useful to us. So these are the two different uh, area of study when it comes to metabolomics. So the typical workflow of metabolomics analysis. So first thing is the detection. So we usually use analytical platforms like mass spectrometry or NMR to detect the um, mass to charge ratio of compounds, and then we get a file in the format of MZML where these data is present. So that is a raw file or the input file, just like we use FastQC for shotgun analysis, that is the input format. So in metabolomics, the file is MZML. So from that, we proceed with the downstream analysis. So the second one is pre-processing and normalization. So in pre-processing, what we do is we do peak detection, alignment, and filtering to build a data metrics where we have the name of the compound and then the intensity, the peak intensity, peak area, or peak length, whatever mm, parameter we want to choose, we can choose, and followed by normalization and all. But in our pipeline, we do normalization with the statistical analysis. And after that, annotation and statistical analysis. There we have two types of statistical analysis. We'll be discussing it when we will be doing meta-analysis, univariate and multivariate statistical methods to find the significantly altered metabolites between the groups. And last and finally, we'll be doing functional and pathway analysis. We can check for the metabolites. Where does it belong, which pathway it belongs to and all. We can check in functional and pathway analysis. And if we want to continue the study, we can do multi-omics integration also, like along with genomics and transcriptomics, we can integrate this data as well to get an overall understanding of how the metabolites works. So this is the typical workflow. Depending on the softwares we are using, it will be altered a little bit, but this is the overall typical workflow of a metabolomics analysis. So here it is a diagram of how it works. We have LCMS spectra, and we do peak profiling or peak calling, and then peak annotation, annotating it with ions and even with the database. And after that, we'll get the data. So we'll clear the data, only take the mm, like we filter out only the data that we want. For example, there would be unknown compounds and all. So if we are not looking for any unknown compounds, we can filter it away and take only the known compounds. And then we do statistical analysis and then functional analysis. So this is the overall workflow. So now tools used in the pipeline. So basically, the most widely used tool is MZMine. So MZMine is a paid version. So if you check in most of the metabolomics research papers, they will be using MZMine. But as it is a paid version, we have taken MS-DIAL, which is an open source software. We can download it freely and use it free of free of cost. So that is why we have chose MS-DIAL. Both work in a similar way, only a little bit of difference, but mostly the parameters and all that, they are the same. So MS-DIAL means mass spectrometry data independent analysis. So it's an open source software for processing mass spectrometry based data. So it supports peak detection. We can detect the peaks, and deconvolution we can do, that is trace overlapping peaks. And then there is retention time alignment and metabolite annotation. Everything we can do inside MS-DIAL itself. It works for both targeted and untargeted metabolomics experiments, like we had discussed before. And also it is compatible with multiple mass spectrometry platforms. So we can handle large data sets. Okay. So this is about the first tool that we'll be using, that is MS Dial. So after MS Dial, we'll be getting the compound name and its intensity. So with that information, we'll be moving on to Metabolomics, where we do the statistical testing. So this is a web-based platform. We don't need to install it or anything. We can just do this in Google. There is a Metabolomics like site. There we go and upload our file. So then we can do the statistical analysis. They can also do visualization, the PCA plot and all that. We don't need to do any coding and all. We just need to give a file. They can visualize all the graphs and all, and we can do univariate and multivariate statistical method testing like t-test, PCA and everything. We can upload also. And there is also option to do pathway enrichment analysis. So all these things you can do it in Metabolomics. And also Metabolomics is one of the most widely used statistical um, testing softwares for metabolomics. So it is very trustworthy. It is mostly used by researchers. Okay. So it acts as an end-to-end platform. There is so much processes that we can do inside Metabolomics, like from statistical findings to functional insight. We can do pathway analysis in Metabolomics. So there are so many things that can be done in Metabolomics. So moving on, we'll be using a data set for our study. So this is the data set that we'll be using. So there is this site that is Metabolites. So from here we can download metabolomics or data that we want to analyze or we want to work with. So there, this is the ID we'll be using. It's an SRS data. So we can just search it. So this is the paper. So there in this paper, like as a summary, they are looking for nicotinic acid from a particular strain of SREs, and they are trying to quantify. So it's basically a targeted analysis. But what we have done is we have done an untargeted version. We are looking for all the compounds that is present. So we haven't particularly looked for nicotinic acid. We try to identify all the compounds that we could find from the sample. So here we can see some descriptions and all. Here this is the overall study information. Here we can see the protocols. So here we have the mass spectrometry uh, information. So from this information, we are taking all the parameters that are essential uh, to do the MS dial uh, mm, to analyze our data in MS dial. So I'll be explaining it later. So here we can see we are using this apparatus. So based on this apparatus, and there are so many ESI ionization. So when we go to MS dial, they ask for these particular things. And also we'll be checking in this particular column, and then we'll be selecting the options there. So this particular box is important. So that's all with the uh, data. And now we need to download the raw data files for the analysis. So for that we can go here, files. So here we can see knockout files. There is knockout 1, 2, and 3, and we can see the wild type files. So 1, 2, and 3. So in our analysis, we have taken the wild types and wild type and the knockout. So to download these files, you have to go and press here, browse files, and it will take to a page like this, and files. And by clicking uh, these links, clicking these links, we can download the knockout one and wild type ones, these three into our file. So after downloading these zipped files, we have to unzip them. Um, the files will be in the downloads directory. There we have to go and unzip them, and then we can see files like .d format. So this .d format is agile and directory based raw files. So there we can see it is not the traditional .mzml type of file. So MZML file usually comes from Illumina platforms like that. So if we are using an agile based library and machine, then the raw data that we'll be getting is in the .d format. So what we have to do is, I think in MZMine we have to convert this into MZML, but in MS-DIAL it will take this .d format directly. So .d format is like a directory. Inside this .d file, there is some some more subdirectory and subfiles. So in MS-DIAL, they'll take this .d files directly and they'll do the analysis. We don't have to change it into MZML. So when we are doing the command line functions, so when I talk about command line functions, command line are there. There are some tools which they use to do command line analysis, but they are very old and they're not like updated and all. Also, people prefer to do it in MZ. Like researchers usually use GUI methods to analyze metabolomics data. So, if we are doing command line version, then we have to convert this .d into MZML using there is a tool that is MS Convert, which we can use for the conversion. So for MS Dial, we don't need to convert it. So that's about the dataset information. And then we have to install the software. So this is a link to install the software. We have to download this zip file, and then we have to extract, and there is this msdial.exe file inside that. So after installing, we need to launch this particular .exe file, and then we can, like, msdial will be ready to use. And one thing we need to download is the database. So this database is required for the annotation, not annotation basically, to the metabolite annotation basically. So we can there will be ADX that are getting annotated, and to identify uh, these peaks belong to which compounds and all, we need to align with particular databases. So yes, sir. Yes, sir, right. Yeah, so the last one I have sent is the database folder. So this is provided by MS Dial itself. So this page, like from this page, we can download all the databases that we need for the annotation and alignment. Here we go to MS Style, Metabolomics, MSP, Spectrum, and all. From here, we have to download the uh, what, .msp files. So we can use whatever database that we wanted. I have downloaded Mass Bank, GNPS. I think so. One from this also. I think it's the MSMS positive one. So basically in untargeted, we can do both positive and negative to check for all the compounds. But as this is a demonstration, I have done only positive analysis. So if you want the negative ones also, you can repeat the steps by giving the
Now, what is this M is positive and negative?
mass spectrometry.
What
Yeah,
stands for positive and negative?
it's the adducts used. So positive adducts are used to get the positively charged metabolites, and negatives are used to catch the negatively charged metabolites.
Okay.
So, so if we are going for untargeted, we would be checking for all the metabolites, right? So then we need to look for both positive and negatives. So here, in this study, they were looking for nicotinic acid. So their metabolite of interest is nicotinic acid, and its accurate mass is M plus H. So it is a positive ion. They have used this adduct to capture the nicotinic acid. So like this, we'll be giving positive adducts to capture all the positively charged metabolites. And if we want negatively charged, so if you're looking for a particular compound but that is negatively charged, then you can give the negatively charged uh, adducts. So that a particular thing will come under, uh, like when we do the analysis, there is an option where we can select. So here they ask for ion mode. So at once we can only select positive ion mode or negative ion mode. So if we are going for untargeted, we have to do the analysis for positive ion mode. Then we get the result, and then we have to go for the negative ion. So we can discuss that further in the demonstration. Yeah, so here I have selected this ESI positive MSMS and GNPS MSMS from the database. Here you can click and download them. You can download as much databases that you want. Uh, uh, like for these two databases, I have used it. Took almost like half an hour for the whole process. So if you select more, it is good, but it will take more time. So accordingly, you can select this GNPS and this ESI. from authentic standards. So these two have
So based on what parameters we have to select these databases?
that is according to our choice. So if we want to use all the databases, we can use. So if we are working with the microbial data, so this is a bacteria, right? So then we can select
Mhm.
GNPS, pain, HILIC. So there are particular databases for bacteria, microorganisms. So these ESI, MSMS, they contain both human and bacteria and all, or like all organisms. So likewise, we can select the database.
So is there anywhere where it is written that which database is for what? Like
Uh,
which database is for bacteria and which is for like human or which is for plant?
uh, that I'm not aware, and I have to check.
Okay. Because suppose I am new, so I
Yeah,
need to know which databases I want to follow.
yeah, yes. There you can check literatures where, mm, yeah, so like this you can check. For example, if you check mass bank, they'll be describing what all data is present in mass bank. So you can check it individually.
Hmm.
Or you can check
Okay.
related papers like related studies with uh, the study that you are trying to do. So if you refer that kind of papers also, you'll be getting some information. So likewise also you can check.
No, from paper I can get it, but there must be somewhere it is. If you can provide that which database is for what, so it would be beneficial for us.
That kind of data I also don't know, ma'am. I have just checked like what all databases have what kind of information, and then I selected two of them. I didn't see any particular
Okay,
paper regarding.
okay. So you went to the paper and then you select.
Yeah, yeah, yes.
Okay.
So these are the requirements that we need to do the analysis. So if you download the sample data and install the software and the database, then tomorrow we can like, do the analysis together. You can try down.